Welcome to the deep dive. If you've ever wondered, how much of your life is really set in stone by your genes, or maybe how much control you actually have over your health,
Right.
strap in. Today, we're really going deep into the field of epigenetics. And this isn't just a science story. It's actually a profound challenge to, well, almost everything we think we know about fate, free will, and even inherited responsibility.
Absolutely. And this deep dive, it's a bit unique because our sources, they really move beyond just the lab bench. They draw heavily from a pretty critical workshop. This was put on by the German Federal Ministry of Education and Research and the Karlsruhe Institute of Technology. They specifically focused on examining the ethical, legal, and social implications, the ELSA, as it's called, of this whole epigenetic revolution.
Right, and that's the mission right there, isn't it? To understand what happens when these, molecular insights jump out of the petri dish and land right in the middle of public policy or intergenerational arguments, maybe even the courtroom. It's really about how this fundamental biology starts shaping our social fabric, our legal structures.
Precisely. And our sources, they talk about epigenetics in this context, not just as a scientific field, but more like a programmatic umbrella term, or even a boundary object, which is an interesting way to put it. Think of it like a conceptual meeting place. It's specific enough scientifically to hold its identity, but it's also open and flexible enough that, really diverse people, molecular biologists, philosophers, lawyers, they can all use it, often with slightly different angles or interpretations. What we'll be focusing on is how this powerful idea forces society to, well, fundamentally rethink things like individual responsibility and the role of the state in governing health.
Okay, so let's start with the basics, the science bit.
Yeah.
Because the terminology can get pretty dense pretty quickly. If the genome is the sort of static underlying blueprint, what exactly is epigenetics?
Right. So epigenetics, it literally means above the genes. And it refers to the regulatory mechanisms, the switches, if you like, that control gene activity. And crucially, they function independently of the actual underlying DNA sequence itself. It's not about changing the letters of the DNA, the A's, T's, C's, and G's. It's all about changing how those letters are read or expressed. You could think of the DNA sequence as the fixed hardware in your computer and the epigenome. Well, that's more like the operating system that decides which programs get run and which ones stay shut down.
That's a really helpful analogy.
Okay.
So when we talk about the epigenome, this whole regulatory system, what are the main mechanisms? What are the key ways this control actually happens?
There are three main players, you could say, and they're distinct, but they definitely interact. First up, you have DNA methylation patterns. You can picture these as tiny chemical caps, usually methyl groups, that get stuck onto the DNA molecule, often onto cytosine bases. When these caps are placed in specific spots, particularly near the start of a gene, they tend to act like a hard-off switch. They silence that gene.
Okay, so methylation is pretty binary, more like on or off. What about the other mechanisms? Are they different?
Yeah, the second one is quite different. It's histone modifications. See, DNA doesn't just float around loose in the cell nucleus. It's actually tightly wound around these spool-like proteins called histones. Now, you can chemically modify these histone spools, maybe add acetyl groups, for example, and these modifications can either loosen or tighten how the DNA is wound. If the DNA is wound really tightly, the cellular machinery can't get access to read the gene, so it's silenced. If it's loosened up, the gene is accessible and can be expressed. So this mechanism is much more like a dimmer switch. It controls the volume of gene expression, not just on or off.
Right, a dimmer switch. Got it. And the third one?
The third mechanism involves various types of RNA expression, specifically things like non-coding RNAs. These molecules don't code for proteins themselves, but they play crucial roles in mediating or interfering with gene functions, sort of fine-tuning the whole process. So the complete set of all this information, the specific methylation patterns across your genome, the state of your histone modifications, all the regulatory RNAs floating around, that's what we call the epigenome. It's really how a single genome, the same DNA blueprint in every cell, can produce such incredibly different cell types. A brain cell ends up so different from a liver cell despite having the same genes.
Okay, that makes sense. No, the basic science isn't brand new, right, Dee? But there's definitely a feeling of an epigenetics boom right now. What's really fueled this acceleration? Was it a particular discovery or more about the tech?
Oh, it was overwhelmingly a technological leap. Primarily, it's down to next-generation sequencing, or NGS. This tech just revolutionized our ability to sequence DNA rapidly and cheaply. But Maybe even more importantly for epigenetics, it gave us high-throughput methods to map all those modification patterns, the methylation, the histone marks, right across the entire genome. This allows researchers to generate these incredibly detailed, high-resolution epigenome reference maps.
And this isn't just happening in isolated labs, right? This sounds like a huge coordinated effort.
Absolutely. It's massive global collaboration. The data generation and sharing, it's largely coordinated through these huge international initiatives. The most prominent one is probably the International Human Epigenome Consortium, or IHE. Their massive goal is basically to map the full epigenomes of hundreds, even thousands of different types of healthy and diseased cells across various tissues and developmental stages. The sheer scale of this effort just confirms the scientific consensus. Understanding the epigenome is absolutely critical for the next wave of medical breakthroughs. Hashtag 1.2, the promises of epigenetic knowledge.
Okay, let's talk about those breakthroughs then. If we can actually map and understand these genetic switches, what are the biggest hopes in medicine? Where could this lead?
Well, the medical potential it's really enormous, especially when you think about tackling those complex, multifactorial diseases that plague Western societies. Things like cancer, Alzheimer's, cardiovascular problems, type 2 diabetes. The real excitement stems from a fundamental difference between the genome and the epigenome. Unlike the genetic code, which is pretty much fixed for life, the epigenetic code is potentially reversible.
Ah, okay, that is a fundamental difference. A gene mutation is usually permanent, right? But an epimutation, maybe you could fix it.
Exactly. That's the core idea. So new therapeutic strategies could involve direct intervention. We could potentially develop drugs that specifically target and change harmful epigenetic marks like abnormal DNA methylation patterns or problematic histone modifications. For example, in some types of cancer, we know that crucial tumor suppressor genes get switched off inappropriately by excess methylation. An epigenetic drug could, theoretically, remove those methyl groups, essentially switching that protective gene back on.
That's fascinating.
Yeah.
But the influence isn't just about developing new drugs, is it? It also seems to connect really strongly to our daily lives, our behaviors. It reinforces this whole lifestyle component, but maybe in a much deeper way than just, eat your vegetables.
Yes, and this is probably the most captivating aspect for many people, and certainly the most socially relevant one. There's really strong evidence confirming that our everyday behaviors, our chronic experiences, they literally leave measurable chemical traces on our epigenome. This includes the obvious things, like our dietary habits, what we eat, what we don't eat, and of course consumption of things like nicotine or alcohol.
But I've seen research hinting it goes even deeper than that, into like social factors, stress.
Absolutely. The epigenome effectively acts as a molecular mirror, reflecting our broader environment, the one outside our bodies. We're talking about the lasting biological effects of things like chronic stress or traumatic experiences. Even social experiences, like the quality of maternal care in early life, whether it's nurturing or neglectful, have been shown to leave epigenetic marks. And of course, specific chemical exposures in our environment. Take high cortisol levels from chronic stress, for instance. It doesn't just make you feel stressed out. It can lead to measurable changes in the methylation patterns of genes involved in regulating the stress response itself. This could potentially hardwire a predisposition towards anxiety or depression later in life.
Wow, so it's like your life story isn't just stored in your brain's memory. Bits of it are actually being written onto your DNA in chemical ink. Hashtag 1.3, the transgenerational component, Lamarckian echoes.
And that leads to the most revolutionary and frankly the most controversial part of the story. If these chemical tags, these epigenetic marks, are stable, how far can they actually travel? Do they just affect us, or?
This is where we bump right into the ghost of Lamarck, isn't it? The old, mostly discredited idea that characteristics acquired during life could be passed down to offspring.
Yes, exactly. Although the mechanisms are completely different from what Lamarck originally proposed, the core idea inheritance of acquired traits does have this unsettling resonance. See, the defining feature of these epigenetic markings, or epimutations as they're sometimes called, is their stability when cells divide through mitosis. That's why, for instance, a liver cell always produces more liver cells. It passes on its epigenetic identity. But here's the kicker. Researchers have shown, quite convincingly in animal models, and with growing evidence in humans, that these epimutations are sometimes inheritable across generations, passed down from parent to child, maybe even further. That's what we call transgenerational epigenetic inheritance.
Okay, wait. So if these marks are caused by environmental factors like diet or stress and then they get passed on, we're talking about my experiences, my behaviors, potentially influencing the health of my children or even grandchildren who haven't even been conceived yet.
That's precisely the implication. And again, unlike genetic mutations, which are generally permanent changes to the DNA sequence, these epimutations are, at least in theory, potentially reversible or erasable. Though, actually erasing them often requires significant cellular events, like the reprogramming that happens during early development.
Right. So what's the classic example? What's the model that really demonstrates this transgenerational effect most clearly?
That would almost certainly be the agouti mouse model. It's cited constantly because it illustrates so powerfully how, let's say, lifestyle choices, or at least environmental inputs like diet, can affect future generations. So these agouti mice, they have a specific gene variant. When this gene is active or unmethylated, it leads to a recognizable syndrome. The mice have yellow fur, they become obese, they're prone to diabetes, and they have a much higher risk of cancer. Basically, they're unhealthy mice.
Okay. And how did researchers manage to influence that outcome epigenetically?
What they did was supplement the mother mouse's diet crucially, before and during her pregnancy, with high levels of specific nutrients that are known to donate methyl groups. Things like methionine, folic acid, vitamin B12, choline, zinc. These are key components for the cell's methylation machinery. This dietary intervention led to a significant increase in DNA methylation, specifically at that agouti gene in her developing offspring.
And the result? What did the baby mice look like?
The resulting pups, even though they were genetically identical to the unhealthy yellow mice, they had the same agouti gene variant, were born completely different. They had healthy, darker brown coats. The increased methylation had effectively silenced that harmful yellow-producing gene variant. But here's the really critical part for the transgenerational discussion. This healthier brown coat phenotype and the underlying protective methylation pattern wasn't just seen in the immediate offspring, the F1 generation. In some studies, it persisted into the grand offspring generation, the F2s, even when the F1 generation ate a normal diet.
Wow. Okay, that changes things. That makes the link between, say, a grandmother's diet during a specific period and her grandchild's potential health outcome feel much more direct, almost tangible, than just thinking about general family habits passed down culturally.
It absolutely does. It creates this almost biological chain of responsibility across generations. And, our sources also point out that research is increasingly exploring links between epigenetics and human traits that were previously assumed to be purely genetic or purely environmental. For instance, there's a relatively younger theory, proposed by Rice and colleagues back in 2012, which tries to connect the origin of homosexuality to specific epigenetic mechanisms operating during fetal development.
Whoa, okay. That immediately pushes epigenetics into some very sensitive territory, cultural debates, identity politics. What exactly did that theory propose? How would that work?
The core idea was that certain epigenetic marks laid down in the womb perhaps to optimize sex-specific traits normally get erased between generations, but sometimes this erasure process might be incomplete. So these leftover marks could be gender-specific, maybe originally intended to enhance fertility or sensitivity to certain traits in the opposite sex parent. For example, imagine an epigenetic mark that increases a mother's sensitivity to male traits. If that mark isn't fully erased and gets passed on to her son, the theory suggests it could potentially influence his sexual orientation towards males.
I see. So an epigenetic mechanism, perhaps evolved for one purpose, like boosting reproductive fitness in one sex, might inadvertently missteer a related trait if it persists into the next generation in the other sex.
Exactly. It's a hypothesis, of course, but it highlights how epigenetics provides mechanisms that challenge those simplistic nature versus nurture dichotomies. It suggests this really complex layer of molecular inheritance that isn't strictly genetic in the traditional sense. We also see clear evidence of epigenetic problems or anomalies arising directly from human interventions like reproductive technologies. Think about in vitro fertilization, IVF, or reproductive cloning, which has been done in animal breeding. Studies consistently show higher rates of epigenetic errors in offspring produced via these methods. This really underscores that manipulating those very early life stages carries significant and perhaps not fully understood risks to the developing epigenome.
Okay, so this whole scientific shift we've been talking about, it really represents nothing less than a major challenge, maybe even an overthrow, of what our sources call extreme genetic determinism, or EGD for short.
That's right. The EGD view really dominated thinking in the late 20th century, especially after the discovery of DNA structure. It basically saw genetic information as holding a total monopoly on determining who we are. The phenotype, your observable traits, your health, your characteristics, was seen as being determined almost solely by your genotype, your DNA sequence. It was almost like a modern version of preformationism, the old idea that a tiny, fully formed human existed in the sperm or egg. In the EGD view, the environment was seen as just, well, nutritional substrate. It provided the building blocks, but it couldn't really alter the destiny already written in the DNA code.
And a classic example of that older, more fatalistic view would be those highly penetrant single gene disorders, right? Like Huntington's disease. If you inherit that specific gene mutation, you will develop the disease. No amount of healthy living or positive thinking is going to stop it.
Exactly. That's the paradigm EGD was built on. But epigenetics fundamentally shatters that kind of fatalism, at least for most traits and common diseases. The new paradigm argues that development isn't predetermined by genes alone. Instead, it's the result of a constant dynamic gene-environment dialogue. The genome doesn't hold all the cards, informationally speaking. Instead, the epigenome, that layer of control within the cell, which is responsive to signals from outside, acts as a kind of mirror of the external environment, as Cooney put it in 2007. This completely reframes our understanding of development, disease risk, even personality.
And probably the most compelling, or at least the most easily understood evidence for this dialogue comes from those famous twin studies, right?
They are absolutely central to the argument against strict genetic determinism. Identical twins, monozygotic twins, start life with essentially 100% identical genotypes. So any differences that emerge between them as they grow older, any phenotypic divergence must be attributed to environmental factors interacting with their genes, likely via the epigenome. There was a landmark study by Frage and colleagues in 2005 that illustrated this just perfectly.
Right. Tell us a bit more about what they found, specifically how things changed over time.
Well, they looked at pairs of identical twins across a wide age range, and they found that when the twins were very young, say around age 3, their epigenetic patterns, specifically DNA methylation and histone acetylation, were remarkably similar, almost indistinguishable. However, as they looked at older twin pairs, these epigenetic patterns had diverged significantly. And crucially, the degree of epigenetic divergence correlated directly with how different their life experiences had been. Twins who had spent more years living apart or had significantly different lifestyles, maybe one smoked and the other didn't, or they had different diets, different levels of stress showed much greater differences in their epigenomes compared to twins who had lived very similar lives. It's really powerful, almost irrefutable proof that the environment contextually determines which genes are active and how active they are. The basic blueprint is fixed, yes, but the instruction manual telling the cell how to use that blueprint is constantly being edited by experience. Hashtag 2.2, the risk of the hidden determinism, VGD.
Okay, so great news. We've achieved this kind of victory over genetic fatalism. We're no longer just prisoners of our DNA blueprint. That sounds incredibly empowering, doesn't it? But our sources identify a pretty significant potential downside, a societal danger lurking in this positive shift. They talk about the paradox of volition, or what they term hidden determinism, VGD. What's that about?
Yeah, this hidden determinism is a really subtle but important point. It arises because, in a way, the pendulum might have swung too far in the other direction. If genes are no longer seen as fixed destiny but as plastic and steerable, the cultural reaction, especially in Western societies, has been to embrace this idea of gene activism. Society, the media, even sometimes public health messaging starts urging people to become the authors of their own luck, to actively steer their genes towards health by choosing the right environment, the right lifestyle. And this is where individual responsibility can very easily, almost tragically, morph into individual guilt.
How does that actually play out, that shift from responsibility to guilt? How might we see it in, say, everyday life or in public discussions about health?
Well, it fundamentally shifts the perceived cause of illness. It moves it away from the biological level, often seen as uncontrollable nature or bad luck, and places it squarely onto the volitional level, the realm of individual action, choice, and willpower. So if you develop a condition like type 2 diabetes, the narrative can subtly shift. It moves from, oh, that's unfortunate, perhaps you had a genetic predisposition, towards something more like, well, why didn't you manage your diet better? Why didn't you control your stress levels? You had the power to steer your genes away from this outcome.
Yeah, I can see how that places a really heavy cognitive and emotional burden on the individual. It sounds exhausting, actually.
It really is. Because this view, this gene activism, it often promotes a rather asymmetrical view of that gene-environment interaction we talked about. It implicitly suggests that there is a desired health-promoting gene activity pattern out there, and it should be achievable if you just make the correct environmental choices. But this completely overlooks the massive structural, social, and economic constraints that drastically limit people's ability to choose their environment. If you live in a food desert with no access to affordable fresh food, if you're working 3 low-wage jobs just to survive, if you're subjected to chronic discrimination or social stress, you simply don't have the autonomy to just choose the right environment to optimize your epigenome.
Right. So epigenetics, which in theory could highlight how social inequality gets under the skin, could instead be twisted into a tool for blaming the victims of that inequality for their poor health outcomes.
That's exactly the risk. The idea of being the sole author of one's own luck, while appealing on the surface, becomes dangerous and potentially harmful when applied to health. Because health, at the end of the day, remains fundamentally unavailable to complete human control. It's incredibly complex, subject to random biological chance, intricate gene-environment interactions we don't fully understand, and those inescapable social and environmental pressures. When this reality asserts itself, when people get sick despite trying their best, the burden of perceived failure falls entirely on the individual. It fosters guilt and shame, rather than leading to practical support or addressing the root causes. So this really forces us to look beyond the individual and consider those broader structural factors. Promoting total individual responsibility for epigenetic health in a society marked by profound inequalities is, well, it's fundamentally unjust.
That whole discussion about shifting responsibility, about blame and guilt, it leads us straight into probably the thorniest ethical jungle in epigenetics. The whole question of intergenerational justice. We're talking about how the choices we make today, what we eat, how stressed we are, the chemicals we're exposed to, might actually affect the epigenetic health of descendants we will literally never meet. And those Överkalix studies from that parish in northern Sweden, they provide a really powerful, almost unnerving, real-world example of this exact moral dilemma.
Yes, the Överkalix studies, first really brought to wide attention by Kadi and colleagues around 2002, they are pretty renowned in this field,
Hello.
partly because they were able to use meticulous genealogical records and parish data from a relatively isolated population going back generations. This allowed them to rigorously track not just family lines, but also historical data on things like harvest quality, indicating periods of feast or famine, and link those ancestral environmental conditions to specific causes of death, like cardiovascular disease or diabetes, in subsequent generations.
And they focus specifically on nutrition during what they call the slow growth period, right? A particularly sensitive window in development. What did they find out about the grandfather's experiences specifically?
That's right. They identified this pre-pubertal slow growth period, roughly between the ages of 9 and 12, as being particularly sensitive for establishing certain transgenerational epigenetic effects, especially transmitted through the male line. What they found was quite striking and gender-specific. If the paternal grandfathers, so the study participants' fathers had experienced periods of abundant food, basically feasts, during their own slow growth period decades earlier, it significantly increased the risk of their grandsons dying from cardiovascular disease and diabetes. Their lifespans were also shorter, on average.
Wow. So the grandfather's good fortune, his abundance, effectively became a health liability for his grandson 2 generations later. And was the opposite also true? Did scarcity have an effect?
It seemed to, yes. Conversely, when the fathers themselves had experienced periods of poor nutrition or food scarcity during their slow growth period, it seemed to have a protective effect on the risk of cardiovascular disease in their direct offspring, the study participants. The interpretation is that the epigenetic response to food scarcity might have been to program the sperm for metabolic thriftiness in the next generation, sort of preparing them physiologically for a world where resources might be scarce. But in a modern world of abundant food, that thrifty programming could become maladaptive.
Okay, this research forces us to confront a really profound and frankly uncomfortable ethical question. Let me try and phrase it directly, as one of the commentators, Philip Bode, did in the source material. If parents know, based on this kind of statistical evidence, that subjecting their own child to, say, mild undernutrition during those first 12 years or so could statistically improve the health outcomes of their potential great-grandchildren, are they morally permitted, or maybe even morally obligated, to do that? To essentially sacrifice some of their own child's well-being for the potential benefit of a future person.
That is the absolute crux of the conflict, isn't it? It pits the tangible welfare and interest of a living, breathing person, your own child, directly against the statistical potential welfare of a future, currently non-existent person. We're navigating what philosophers call the complex ethics of possible people, and our traditional moral frameworks, which strongly prioritize the duties we owe to actual living individuals, start to feel really strained and maybe inadequate when faced with these kinds of transgenerational trade-offs. Hashtag 3.2, the ethics of possible people and non-existence.
Right. Let's try and unpack some of the philosophical tools or frameworks that people use to grapple with these possible people. Anja Karnine, for instance, wrote about the moral protection of the unborn. How does her argument fit in?
Karnine's position, as presented in our sources, is quite nuanced. She argues that born humans, actual persons, receive full moral protection and status. Unborn life fetuses, embryos, doesn't automatically have that same status, partly because of its existential dependence on the mother. However, she strongly asserts that unborn life should still be treated as if it will become a person. This leads her to argue against arbitrary artificial manipulation of embryos, for example. It creates a kind of prospective protective status. The idea is rooted in the notion that if a person is eventually born, they should have a kind of retrospective claim that their embryonic form wasn't treated merely as a thing, that its potential personhood was respected.
Okay, but the moral calculus gets even trickier when we bring in the concept of non-existence itself. Particularly Jeff McMahon's challenging idea from back in 1986, known as the complaint condition.
Yes, McMahon's argument is famously counterintuitive but philosophically powerful. It directly tackles the question: can a person logically complain about their own existence if the only alternative for them was simply never having existed at all? The traditional view, often based on a simple principle of beneficence, tends to assume that being born, having any kind of life, is always preferable to non-existence. Therefore, bringing someone into existence can't really be considered a harm to them.
Right. That feels like the default assumption. But McMahon pushes back hard against that, doesn't he?
He fundamentally challenges that assumption, the idea that every life is intrinsically better than no life at all. He argues that if a life is foreseeably going to be so severely impaired or burdened, perhaps due to inherited health defects, maybe even defects caused epigenetically by a parent's actions or environment, that it is reasonably judged to be worth not living, then bringing that person into existence can constitute a legitimate harm to that person.
Okay, hold on. So a person who is alive but is living a life of extreme, perhaps avoidable suffering, can retrospectively complain about the fact that they were born, even though the only alternative for that specific person was nothingness. That still feels very difficult to wrap one's head around.
It is difficult, absolutely. But the philosophical point McMahon makes is that the harm isn't somehow erased or negated just because the alternative was non-existence. If you, as an actual living person, are experiencing severe, unavoidable suffering that could have been prevented before you were conceived or born, McMahon argues you do have grounds for a complaint against those who allowed your existence in that impaired state. And epigenetics makes this previously abstract philosophical debate incredibly immediate and concrete. Because now we're identifying actual biological mechanisms through which parental behavior or environmental exposures can directly contribute to potentially severe and maybe avoidable health impairments in future generations. Hashtag 3.3 theological and societal responsibility, the original sin analogy.
Shifting gears slightly, but staying in the realm of moral frameworks, our sources actually draw a structural parallel between the idea of inherited epigenetic predispositions or defects and the theological concept of original sin. Now, that sounds like a pretty heavy comparison to make.
It is a heavy comparison, definitely. But Harold Mattern, writing in 2016, used it quite specifically to frame a particular social problem, not necessarily to make a direct theological claim. If you strip away the specific religious narratives and moralistic interpretations, the core structural idea behind the concept of original sin, Matter argues, reflects something real. The unhintergebor, that's a German word meaning something like unavoidable, inescapable, something you can't get behind, the unavoidable and persistent nature of certain individual behavioral dispositions, defects, or weaknesses that seem to be passed down across generations. It's about acknowledging that we are often born into a state of structural imperfection, carrying burdens or vulnerabilities that are not, in any simple sense, our own fault.
Okay, so the analogy isn't really about blaming Adam and Eve for everything. It's more about recognizing that our starting point in life, our biological and maybe even social predispositions, is inherited, largely unavoidable, and significantly influences our capacity to act freely or achieve certain outcomes, like perfect health.
Exactly. It's about that inherited structural constraint. And the ethical reflection that Mattern and others draw from this structural similarity leads to a really crucial policy or societal mandate: demoralization. Given the radical complexity of how these dispositions originate, this intricate mix of inherited epigenetic patterns, social stress, environmental toxin exposures, random biological chance, maybe some genetic factors too, we absolutely must refrain from moralizing specific resulting behaviors or illnesses.
Meaning we need to stop framing things like, say, obesity primarily as a moral failing of the individual, or judging someone's anxiety as just personal weakness, especially when epigenetics is suggesting these conditions might be partly programmed by factors well beyond their immediate control, maybe even factors from previous generations.
Precisely. This demoralization should apply across the board, whether we're talking about metabolic conditions like diabetes, mental health challenges, or even things the media sometimes sensationalizes, like non-normative sexual preferences potentially linked to epigenetic factors. The sheer complexity demands that we move away from simplistic individual moral judgment.
So if we demoralize, what's the practical takeaway? What does this mean for how we think about responsibility in this new epigenetic framework?
Well, the conclusion is that responsibility needs to be both clarified and simultaneously significantly moderated. The idea of total responsibility, either for oneself or for others, especially future generations, is simply not achievable for human beings, given all these complexities. It's an illusion. Therefore, the focus really needs to shift decisively. We need to move away from assigning individual blame and guilt and move towards building pragmatic structural support and enhancing institutional infrastructure. What does that look like? It means things like better public education about heritability and risk, not to scare people, but to inform them. It means stronger family support systems. And critically, it means actively addressing the underlying social determinants of health, poverty, inequality, environmental pollution, the very factors that negatively program the epigenome in the first place across populations.
Okay, let's pivot now to governance and regulation. Because the very same flexibility of the epigenetic code, the thing we hailed earlier as being so promising for potential therapies, also makes it acutely vulnerable, doesn't it? Especially to environmental chemicals. Unlike the relatively stable genetic code, the epigenetic code seems like it can be more easily altered by common chemicals. We encounter things like bisphenol A, BPA from plastics, benzene from pollution, components of nicotine smoke. And if these alterations happen in germ cells, in sperm or eggs, they could potentially lead to those transgenerational effects we discussed.
Yes, and this vulnerability brings us face to face with a truly monumental public health and regulatory challenge: the problem of endocrine disrupting chemicals, or EDCs. These chemicals basically throw a wrench into the works of how modern chemical risk assessment is typically done. Traditional toxicology for decades has largely relied on a core assumption: the dose makes the poison. There's a simple dose-response relationship. The higher the exposure, the greater the harm. And below a certain threshold dose, the chemical is considered safe.
Right, that's the standard approach. But EDCs, they don't play by those rules, do they?
No, they absolutely do not. EDCs often exhibit what's called non-monotonic dose responses, meaning sometimes a low dose can have a greater effect than a high dose. But even more fundamentally, their effects show a critical time dependency, not just dose dependency. Exposure to even a tiny amount of an EDC during a specific sensitive developmental window, when crucial epigenetic programming is happening rapidly, can potentially cause far more significant and lasting harm than exposure to a much higher dose later in life, outside that critical window.
Yeah, and what are these critical windows? When is the epigenome most vulnerable?
They are concentrated during periods of intense hormonal activity and rapid cell differentiation. So we're talking about early embryonic and fetal development, infancy, childhood, and particularly puberty. Exposure to EDCs during these times can significantly increase the risk for various adult diseases, metabolic disorders, reproductive problems, certain cancers, neurodevelopmental issues, often years or even decades down the line. Furthermore, EDCs often don't cause classical tissue damage, what German law calls Schädigung, like physically breaking a DNA strand or killing cells. Instead, they cause misdirection or missignaling, what's termed Bildstörung. They subtly interfere with normal hormonal signaling pathways, confusing the body's developmental or metabolic instructions.
Okay, so it's not outright damage, but more like giving the body the wrong instructions at a critical time, which sounds much harder to detect and regulate.
Exactly. It's much harder. And to make matters even more complicated, their effects are often non-monocausal. It's rarely just one chemical acting alone. Their cumulative exposures add up over time, and they can be synergistic. The combined effect of multiple EDCs might be greater than the sum of their individual effects. We're all exposed to a complex cocktail of hundreds of these compounds daily through food, water, air, consumer products. This makes isolating a clear, single cause and effect relationship, which is what traditional regulatory models rely on, virtually impossible. That's why existing frameworks like the EU's REACH, Registration, Evaluation, Authorization, and Restriction of Chemicals, or CLP, Classification, Labeling, and Packaging Regulations, are frequently criticized as being inadequate for identifying and properly regulating the risks posed by EDCs.
And the absolute poster child for this regulatory struggle, this mismatch between the science and the policy, has to be bisphenol A, BPA, right?
Oh, absolutely. BPA is pervasive. It's been used for decades in polycarbonate plastics like baby bottles, reusable water bottles, the epoxy resins lining food and drink cans, even on thermal paper used for cash register receipts. Studies, including large-scale biomonitoring studies in places like Germany, have found detectable levels of BPA in the urine of something like 99% of children tested. It's everywhere. The huge controversy revolves around what constitutes a safe level of exposure, the tolerable daily intake, or TDI. Numerous independent toxicological studies, often using animal models, have repeatedly demonstrated harmful effects, particularly on hormonal systems, metabolism, and neurodevelopment at exposure levels far below the officially established TDI set by regulatory agencies like the European Food Safety Authority, EFS.
And this scientific discrepancy, this debate about safety at low doses, has actually led to some countries breaking ranks, hasn't it?
Yes, it has. Faced with the mounting scientific evidence suggesting harm, especially to vulnerable populations like infants and pregnant women, and despite the official European regulatory stance maintaining the TDI was safe, some individual EU member states, notably France and Sweden, decided to implement stricter national measures. They banned BPA in infant feeding bottles initially, and later in all food contact materials. This really highlights a significant regulatory lag. Our existing risk assessment paradigms, which were largely built to deal with classical high-dose industrial poisons, seem to be failing to adequately protect the public from the different kinds of risks posed by these modern, low-dose, hormonally active, and potentially epigenetically active chemical disruptors. Hashtag tag 4.2 legal and state protection obligations.
Okay, so if these chemical exposures, or maybe even severe stress or malnutrition, can cause these epigenetic changes that demonstrably lead to negative health outcomes, well, then this clearly becomes a matter for the legal system for accountability. What are the potential implications under civil tort law, for example?
Right. Under general principles of civil tort law, the law dealing with wrongful acts causing harm and epigenetically induced change that leads to a detrimental deviation from normal bodily function or development could potentially be legally recognized as a health injury. If you can prove that someone's action or negligence caused this epigenetic harm, which in turn led to a recognized health problem, this could, in theory, justify a legal claim for damages compensation for medical costs, lost earnings, etc., and possibly also for pain and suffering. And importantly, tort law in many jurisdictions doesn't just protect living people. It often extends protection to the unborn child, the nascituras, from the moment of conception, provided that the injurious action can be demonstrably and causally linked to harm suffered by the embryo or fetus, assuming the child is later born alive.
That sounds like establishing causation, proving the specific exposure led to that specific epigenetic change which caused the specific health problem. That sounds like an incredibly high bar, a massive burden of proof for someone bringing a lawsuit.
Oh, it absolutely is. Proving causation in these complex, multifactorial, long latency scenarios is extraordinarily difficult, legally and scientifically. But the mere potential for such claims is significant. It opens up new theoretical avenues for legal actions against polluters or manufacturers of harmful products. And beyond that, this growing knowledge about epigenetic risks also directly impacts the duties and potential liabilities of the medical profession.
How so? How does knowing about epigenetics change the standard of care for, say, a doctor treating a pregnant woman?
Well, it potentially creates a heightened duty of care under the principles of medical malpractice law, or Arzthaftung, as it's termed in German law. Doctors generally have a professional obligation to be aware of and implement scientifically secured findings relevant to their practice. So, if there's established scientific consensus about certain risk factors known to cause harmful epigenetic changes during pregnancy, for example, the impacts of chronic, unmitigated maternal stress, severe nutritional deficiencies, or heavy nicotine exposure, then a physician arguably has a duty to recognize these risks in their patient, advise them accordingly, and perhaps recommend interventions. If a doctor were to negligently fail to identify or address these known epigenetic risk factors and the child subsequently suffers developmental damages or health problems demonstrably linked to those factors, it's conceivable that this failure could constitute a breach of professional duty, a form of medical error leading to liability. Epigenetic harm is starting to be recognized as a potential category of developmental disorder that a reasonably prudent physician should be aware of and seek to mitigate.
Okay, so we have potential liability for polluters, for doctors. What about the state itself? Does the government have a role, a legal obligation, to protect children from epigenetic harm? Could evidence of harmful epigenetic changes actually lead to state intervention in family life?
That's a really critical and sensitive question. Yes, in most constitutional democracies, the state is considered to have a fundamental protection obligation, a Schutzpflicht, for the life and health of all its citizens, including, importantly, the unborn, nascituris, and born children. So, theoretically, if there were clear and convincing evidence that severe parental neglect, maybe chronic malnutrition, exposure to toxins in the home, or subjecting a child to constant severe unmanaged stress was causing demonstrable harmful epigenetic changes with likely long-term health consequences, this could potentially serve as a key indicator, maybe even justification, for intervention by child protection services like the youth welfare offices. However, this is arguably one of the most complex legal and ethical tightropes to walk. State intervention into family life requires an extremely high threshold of proof. You need a high degree of certainty that significant harm is actually occurring or is imminent, and that the state's intervention would genuinely be beneficial for the child's welfare, carefully balancing the child's right to protection against the parent's fundamental rights to autonomy in raising their child. It's fraught with difficulty.
And there's one specific, highly debated policy area where this epigenetic lens has been applied very directly, raising some really uncomfortable questions. Institutional childcare for very young children. Toddlers.
Yes, that's the whole U3 childcare debate referring to care for children under the age of 3. This has become a flashpoint, politically and ethically, precisely because of emerging findings related to stress epigenetics. Several studies, particularly some conducted in Germany looking at cortisol levels, a key stress hormone, have found significantly elevated cortisol indicating chronic stress in a substantial minority, sometimes reported as up to 22%, of toddlers aged 1 to 3 who are attending institutional daycare settings. These elevated levels were often correlated with factors like large group sizes, low staff-to-child ratios, or high staff turnover, basically, indicators of lower quality, potentially more stressful care environments.
And high chronic cortisol in early life. That's exactly the kind of signal that can lead to negative epigenetic programming of the stress response system, potentially lasting a lifetime.
Exactly. The biological mechanisms are plausible. High cortisol can influence methylation patterns and histone modifications in genes that regulate the HPA axis, the body's central stress response system. This early life programming can potentially lead to a hyperreactive or poorly regulated stress response later in life, increasing vulnerability to anxiety, depression, metabolic problems, and cardiovascular disease. So this research raises the deeply uncomfortable legal and ethical question. Is the current state of publicly funded U3 childcare, particularly where it might be under-resourced or poorly managed, actually constituting a form of state-supported health injury for a significant number of young children by inducing harmful epigenetic changes? The ethical implication, argued strongly in our source material, is that this evidence places a moral and perhaps even legal obligation on the state to ensure much higher quality standards in early childcare. This means providing substantially greater financial resources, mandating smaller group sizes, ensuring better staff training and stability, all aimed at mitigating chronic stress and creating a supportive environment that fosters optimal epigenetic development during this critically sensitive window. The argument is that the long-term societal cost of not doing this, of allowing adverse epigenetic programming early in life, likely far outweighs the short-term cost of investing properly in high-quality early care. Hashtag TAG 4.3, the public and the Calling Rouge dilemma.
Okay, finally, let's zoom out and look at how the wider public is actually receiving and making sense of this revolutionary and sometimes quite alarming science. Our sources suggest that when ordinary citizens engage with epigenetics, maybe through media reports or public discussions, they tend to latch onto the idea of Eigenverantwortung, that German term for self-responsibility. They get excited about applying this knowledge to their own daily lives. The media often frames the genome as newly flexible, something you can manage.
Yes, that focus on self-responsibility and immediate applicability is a very common reaction. It's understandable the idea that you can take control of your genetic destiny is inherently appealing. But this public discourse, especially in the early stages of the technology, runs headlong into a classic problem in technology assessment and governance, something known as the Collingridge dilemma.
The Collingridge dilemma. Okay, what exactly is that?
It basically articulates a frustrating paradox about timing and control when it comes to new technologies. The dilemma goes like this. When a technology is very new and its societal impacts are still unclear, it's relatively easy to change its direction, to regulate it, to steer its development. But precisely because the impacts aren't clear yet, there's often little societal motivation or data to do so effectively. Then later on, when the technology becomes more widespread and its consequences, both good and bad, become clearer, it's often already deeply embedded in the economy, in social practices, in infrastructure. At that point, it becomes incredibly difficult, maybe even impossible, to significantly change its course or regulate it effectively. It's become too late to steer.
Okay, I see. So applying that to epigenetics, if we try to have a big public debate about the ethics and governance of epigenetics now, when many of the applications are still speculative or only in the lab, people might find it too abstract, too removed from their daily lives to really engage deeply. They don't see the urgency. But if we wait until, say, there are widespread commercial epigenetic tests promising to predict your disease risk based on your lifestyle, or maybe even drugs designed to correct epigenetic marks, by then the industry might be established, public expectations set, and it could be much harder to implement fair, equitable governance structures or regulations.
That's precisely the dilemma. Because concrete mass-market applications of epigenetics are still relatively scarce, even though the research is booming, public interest in debating abstract research agendas or potential long-term societal impacts tends to be quite low. This creates a real risk of what's called artificiality in public participation exercises or citizen dialogues about epigenetics. People aren't genuinely invested or motivated to grapple with the complex ethical trade-offs until the technology is already starting to directly affect their lives, their health choices, or the legal landscape.
So given this dilemma, how should scientists, policymakers, and maybe even the media navigate this? How can we foster responsible development and have a meaningful public conversation before it's too late?
Well, this really underscores the critical need for approaches like responsible research and innovation, or RRI. It's not enough for scientists to just do the research and publish the results in journals. Scientists in particular need to be much more aware that simply publishing what they see as sober research results, like the agouti mouse study showing diet affects grand offspring, or studies linking stress to transgenerational effects inherently shapes public perception and discourse, often in ways that unintentionally fuel that potentially problematic narrative of total individual control and responsibility. The goal of an RI approach is to proactively monitor and try to guide this public discourse, to foster a more nuanced, constructive dialogue that focuses not just on individual empowerment, but also critically on issues of justice, social inequality, environmental responsibility, and systemic risk reduction. It's about trying to ensure that the trajectory of epigenetic research and its applications aligns better with broader societal needs and values, rather than just empowering the already privileged individuals who can afford to buy the right environment or the latest epigenetic fix. Hashtag tag outro.
Well, this deep dive into epigenetics has certainly been eye-opening. It really illuminates these profound connections, doesn't it? Between the microscopic world of molecules on our DNA and the macro structures of our society, our laws, our ethics. It reveals not just this immense potential for new therapies, but also this incredibly complex, maybe even heavy, burden of responsibility that this knowledge seems to place on all of us. We find ourselves biologically linked, not just to our ancestors' wealth or social status, but maybe to their specific diets, their stresses, their exposures. And in turn, we realize we hold this measurable biological influence over the health and well-being of future generations, including people who, right now, don't even exist.
Absolutely. And the crucial takeaway, I think, if we want to navigate this epigenetic revolution ethically and justly, is the absolute necessity of genuine transdisciplinary cooperation. We need biologists talking to ethicists, talking to lawyers, talking to sociologists, talking to policymakers, and importantly, talking to the public. Epigenetics fundamentally demands that we move beyond thinking solely in terms of individualized prevention, personal lifestyle choices, and willpower. That's part of the picture, but it's far from the whole story. The real challenge, the societal challenge, is how we translate these intricate molecular insights into effective structural policies. Policies that actively address the pervasive systemic issues that we know shape health outcomes epigenetically across entire populations. Things like social inequality, exposure to environmental toxins, equality of early life support systems, access to healthy food, chronic stress related to poverty or discrimination. These are the big levers that epigenetically pattern health across generations.
That structural perspective, thinking about those big levers, leads us perfectly into one final provocative thought for you, our listener, to perhaps explore further as you continue your own learning journey? It's a question that touches on much of what we discussed. If we accept the evidence suggesting that the chronic stress potentially induced by poorly managed, under-resourced early childhood institutional care can epigenetically harm a child, possibly resulting in lifelong predispositions to physical and mental health problems, then does the traditional concept of personal freedom and parental autonomy, specifically the parent's right to choose the care environment for their child, or perhaps the state's decision on funding levels, need to be fundamentally reevaluated? Should the state perhaps be granted greater authority, maybe even an obligation, to intervene more strongly to ensure every child's optimal epigenetic starting conditions, even if that means potentially limiting parental choice or significantly increasing public spending? At what point does the child's right to a healthy epigenetic foundation potentially outweigh the autonomy of the parent or the fiscal constraints of the state? That's a really tough tension, something to think about as you look at the world around you through this new epigenetic lens.